mRNA vaccines. In order to understand what this is all about, you're going to need a little bit of knowledge. Hi, my name is Charles. Now, what I'm going to do today is just pass on a little information about this new vaccine. I think it would be helpful to provide some background information. My purpose is to pass on my understanding of the immune response to vaccinations, the basics of cellular biology, and the difference between customary vaccination and the relatively new COVID-19 mRNA vaccination. First, my disclaimer. I am not a virologist or an immunologist. I do not hold a doctorate in any type of healthcare occupation. I have, however, taught physiology and anatomy at 2 different colleges, albeit just the basics, but still just enough to maybe help someone out there understand just a little more about what we're talking about. Now, the science behind vaccinations was discovered when a doctor noticed that milkmaids got cowpox, became immune to smallpox. So he ran an experiment. He took the active cowpox out of these maids, and by the way, this is the grandfather of active immunization that we use today. Today, we introduce a weakened or deactivated virus into the body to invoke an immune response. So back to the story. The doctor rubbed the active cowpox into a cut on a boy's arm. About 6 weeks later, he injected smallpox into the boy. The boy did develop cowpox, which is the mild disease, but he did not contract the deadly smallpox. Thus, the name vaccination means from cows. Now, our vaccination technology is about 200 years old. But we're going to talk about this newer science. First, we need to know a little bit about cellular structure. Cells are the tiniest units of life that carry the genetic code necessary for life. The code is stored in a long double helix string called DNA. The string is made up of multiple components, but the genetic code for life, the DNA, is inside specialized structures in the cell called the nucleus. It's only one nucleus. The key to life is cellular duplication. This is where new cells are made by exactly duplicating the original cell by cell divisions called mitosis. We have assigned designated names to major pieces of DNA strands. They are adenine, thymine, guanine, and cytosine. They combine in different but exact patterns and are designated by the letters A, T, G, C. Now, A only combines with T and G only combines with C. The genetic code for humans is over 3 billion letters long, and it's called the genome. It's just a bunch of pairs of genes, basically. And viruses have codes too, but they're much shorter. The COVID-19 is about 30,000 letters long, 100,000 times shorter than the human DNA. Speaking of 30,000, interesting enough, the human genome is composed of about 30,000 pairs of genes. But in short, DNA holds the recipe for cellular growth by giving instruction for proteins, which is what we'll investigate, and it also passes down inherited information, not for this discussion. So let's shift gears to the immune response. There are several different responses, but I'm only going to give one scenario, the cellular response, instead of the antibody response. Viruses get in a cell and they disrupt all types of good stuff that the cell is trying to do to sustain life. Viruses like to hijack a cell and use it for its nefarious purpose. But when a virus invades a cell, white blood cells called T lymphocytes, these are T cells, recognize the foreign protein, this is called an antigen of the virus, and the T cells prepare to fight the invaders. Infected cells are gobbled up by cells called phagocytes and bring the invader in contact with the T cells. T cells then multiply into killer T cells and memory T cells. Killer T cells target the infected cells by injecting toxic proteins to kill the cell. The memory T cells protect against future invaders of the same kind. Now let's talk more about the DNA. DNA, it's a double helix, twisted ladder type looking thing. It's joined by pairs of nucleotides, and these molecules, these letters that join these rungs, are unwound and then detached in the middle of this ladder. It's kind of like a ladder like this, and it opens up to these nucleotide bases. And what happens, it produces messenger RNA or mRNA and carries the protein building information outside of the nucleus of the cell, but still inside the cell, which is called the cytoplasm. And another structure in the cell, cytoplasm, called ribosome, moves along the messenger in mRNA because it has the code. It translates the information and grabs amino acids to make the instructed protein. Knowing a little bit about mRNA is going to be important next when we are discussing the mRNA vaccine. These new types of vaccine take advantage of using mRNA to give information to build a new protein. Researchers have found out early that introducing mRNA did not work well because mRNA is so fragile. It was questionable about its immune response, and there was some inflammation associated. So researchers learned to wrap mRNA in a bubble made of lipid nanoparticles to prevent destruction of the mRNA that is too fragile and susceptible to enzymatic destruction after the injection. Plus, they gained knowledge about the biology and the chemistry and improved the effectiveness and the stability of what they were doing. These oily bubbles then collide into the cell and fuse to them, releasing the mRNA into the cytoplasm. So for the COVID-19 vaccine, this mRNA has a genetic code that is exactly a piece of the virus, and the ribosomes read this mRNA and build the protein spike. This spike migrates to the surface of the cell. The immune system sees this protein spike as an invader and invokes the immune response and starts building antibodies to target the protein. Now also, when the vaccinated cell dies, there are fragments that are left on the surface and invokes the response and helps corral other immune cells like B cells to fight the infection. And interestingly enough, the team who led the development of this mRNA process include a major contributor to the technology for Moderna vaccine, the most effective and the most stable vaccine. This contributor's name is Dr. Kizzmekia Corbett. She is an African-American lady. You're welcome. Also, remember the part about the DNA code for life is copied inside the nucleus of the cell? This is important because mRNA that give instruction to produce proteins happens outside the nucleus. Therefore, there is no way for genetic mutations to occur as people think with the introduction of mRNA. Also, this fragment of genetic code transmitted by mRNA is not live, weakened, or deactivated. So, you cannot introduce the infection into the body. So then you say, well, what about the long-term protection? Well, verdict is out. It is too early to tell exactly how long the vaccine will provide protection. It's possible that months after the vaccination, the number of antibodies, the killer T cells, will drop, and the immune system may also contain specialized cells called memory B cells and memory T cells that might retained the information about COVID-19 for many years, but we don't know. Only time is going to tell. So we will see. Thanks for your attention.